Hi, everyone. Welcome to Chapter 17. The chapter deals with processes and procedures for identifying and diagnosing different microbial infections. Specifically, we'll be talking about the phenotypic or observable, the genetic or based on nucleic acid, and also the biochemical tests that study metabolism of different microbes as three useful categories of tools to understand and discuss the infections of the human body. So let's get in first by starting to define what these different categories are. So as we mentioned, we have the phenotypic category. And when we talk about phenotypic, phenotype arises from the properties of a cell that are determined by its DNA composition. So obviously, our genetic material governs our physical appearance. So think of phenotypic as physical appearance. These are things that we can observe. So for instance, we can look at microscopic images of the microbe, whether it is stained or unstained specimens, excuse me, shape, size, which we know morphology, but then also the different reactions from things like gram staining, acid fast staining, or endospore staining. We also can look at things macroscopically, morphology. We can grow them on a petri dish or a slant tube on agar. We can look at how the colonies look, their texture, their size, their color, the growth requirements. And then lastly, we have the biochemical characteristics. These are things like the catalase tests, the coagulase tests, whether they are aerobic or anaerobic. So we're able to look at different metabolic pathways. What do they use for nutrients? How do they grow? What's the environment that they prefer best to survive on? So phenotypic is physical appearance. These are traits and things that we're able to see. We also have the genotypic, or you can think of geno as genetic. Genetic techniques, first of all, are incredibly specific. And what we're doing is we are amplifying a very specific region of genetic material. And it only requires very small or minute amounts to do this. So these are molecular methods that are really useful for identifying microbes because all we need is their DNA. There's no need to grow these on laboratory media. There's absolutely no culturing necessary. We can simply break open the cells, extract very minute or small amounts of genetic material, and amplify it using different tools. We often utilize these things called DNA probes, which are these short segments of DNA that actually attach to genetic material. And it's really useful, DNA probes, for us to be able to identify organisms that are grown in pure culture. So these are incredibly precise. They're often automated, so it results in very quick, often within a few hours, results of the outcome. And then finally, we have the immunologic. So this is the study of immune cells. For instance, we refer to things like serology, which is the study of the components of the serum, looking specifically at things like your antibodies. So oftentimes when we talk about immunologic studies, we're talking about antibody antigen reactions. And we'll see that this is incredibly specific. We often refer to this as a lock and key technique because you need a very specific antibody protein to match to a foreign substance or what we refer to as an antigen. And antigens can be anything from a bacterial cell to a dust particle that could result in an allergic reaction. So, specimen collection, obviously, if we're talking about obtaining, characterizing, and diagnosing infections, the first piece to it as future healthcare providers is the collection process. So this really requires sampling from different body regions or fluids from the body, looking for infectious agents. It is really important to make sure that aseptic procedures are always used. We do not want to falsely inoculate a once sterile sample with a microbe due to improper handling. Also, you need to be very cautious about things like storing it in the proper heat or refrigeration. If you create a microbial sample and leave it out on the counter overnight, chances are you're going to get inaccurate results because the microbe is going to grow in that media to very high numbers, giving a very, very false positive. So, results of our specimen collection depend on how we handle, collect, and store the specimen. So, we always want to make sure that we're wearing gloves, we are appropriately applying the technique, whether it's a swab like you see here, and we're also making sure that things like the tamper evident seals are in place. And also the swab is within its plastic case after the specimen is collected. Oftentimes there's a medium that's in here to keep the organism alive while in the testing process. Things like clean catch urine samples, sputum samples, we often talk about. So, a sputum sample is often a mucus sample that's coughed up from the lower respiratory system. Clean catch urine means simply that. We shed off any of the microbes that are in the outer portions of the genitalia and the urinary tract, and we collect a very clean sample of urine for testing. So here are some various guidelines for different types of specimen collections. So obviously, things like an anaerobic culture, we need to make sure have never been exposed to air. Cerebral spinal fluid is often removed by a lumbar puncture, a very long needle that's inserted between the vertebrae. Feces, again, requires samples that are held over 1 hour to be refrigerated. So a lot of this you're going to see requires specific transportation and refrigeration. We've already mentioned urine. Again, it's about cleaning the external genitalia. It is a process of urinating for a short time, followed by the collection to make sure that we are actually getting a clean catch sample. So, what are some of the different laboratory techniques that we are going to utilize once a sample is collected? Well, first of all, we're talking most times about direct tests, and direct testing includes things like the microscopic staining, which we'll talk about, microscopy, and then the macroscopic, which is a lot of tests that we can actually see with the naked eye. We can also do cultures. We can grow them on different forms of media. We can do the genetic sampling to look at some different tests that way as well. And there's really two categories of tests. There's the initial test, which we call the presumptive test, and then there are also confirmatory tests to finalize our initial prognosis. So when we talk about direct testing, let's go through some of the various pieces in a little more detail. The gram stain, obviously, we have mentioned, staining is one of the first tests that any microbiologist will do to start the process of identifying an unknown microbe. We want to understand what its cell wall is, whether it's gram positive or gram negative, which is going to tell us a lot about its peptidoglycan structure. Remember, gram stains and acid-fast stains are differential stains, meaning it allows us to distinguish between bacterial cell types. So the gram stain is often uses a series of stains. We know the stains there start with crystal violet, with the mordant to fix the iodine to fix the crystal violet, followed by an alcohol decolorizer, and then the counter stain, which is safranin. Gram stains will give us either a purple result for a gram positive or a pink result for a gram negative cell. Acid-fast staining, on the other hand, looks for the presence of a very specific bacteria, Mycobacterium. And this utilizes a series of stains. Carbol-fuchsin is the primary stain. It's kind of a magenta purple color, followed by a acid alcohol, which is used to remove the stain, initial primary stain from the cells. And then finally, methylene blue is the counter stain. So what you're going to get in an acid-fast stain is any Mycobacterium are going to stain purplish pink in color from the carbol-fuchsin. Any other cell that's non-acid-fast or non-Mycobacterium is going to stain blue in color. So you're going to get pink cells against a blue background of other cells. We can also utilize antibodies that have a fluorescent tag to them, and we can stain the cells with that fluorescent tag to study them as we expose them to UV light. They will glow and kind of show us where they are located. This is often called fluorescent microscopy. Gene probes, as we mentioned, those DNA probes attach to a very specific site on the DNA molecule. So remember that DNA is made of a series of what we call bases, letters, A's, T's, C's, and G's. And they match together. So two strains will adhere based on their similarities. So an A will always match to a T, and a C will always match to a G. So the probes are designed to go in and attach to very specific sequences of DNA. And we can attach different types of tags, fluorescent tags, to those probes so we can understand if that particular gene exists on that DNA sample. And then we can also do what's known as fluorescent in situ hybridization testing. Hybridization really involves two strands of DNA that are similar. So if you are from the same species, your DNA is going to be incredibly similar in nature. Two different species, so a bat and a human, for instance, are going to have very different DNA sequences. So their hybridization or their ability to come together is not going to be as common. The macroscopic side includes things like direct antigen testing. So looking for the ability of an antigen and antibody to adhere and come together, forming this kind of clumping reaction. We can type DNA, and we can also do a series of biochemical tests to identify a pathogen. Culturing includes things such as biochemical tests. Does it have a catalase enzyme? Does it have the coagulase enzyme? What type of oxygen sensitivity does it have? Serotyping. We can look at the interaction of the proteins or antibodies in the serum. Sequencing the DNA or RNA to figure out what the genetic codes are. PCR, which stands for polymerase chain reaction. PCR is a test that basically takes a segment of DNA and amplifies it or makes copies, millions of copies of it. So it is like a molecular Xerox machine. Phage typing. Phage typing is a really important test because it allows us to distinguish between bacterial strains. We can inoculate a specimen within an animal, and we can also look at sensitivity of microbes to particular antimicrobial agents. So for the patient, it requires us to provide a series of tests and to examine the patient for their signs and symptoms. We also talk a lot about the difference between in vivo and in vitro. In vitro tests are tests that are done in a test tube or on lab media, In vivo tests are done within a patient. It's something that is done to a living organism. So that's important to understand. And then obviously we also have the immunologic and serological tests, things like antibody titer. We can take some of the serum and look for the levels of titer for different antibodies. Obviously, if you've not received a vaccine, but your titer levels are high, that is symbolic that an infection is present in the body. And so, obviously, when we continue on and we talk about the direct examination methods, we have the microscopy specifically around the stains. And when we talk about stains, again, we're talking about looking for and identifying the morphology. So we know it's going to allow us to see things like the bacilli or the cylindrical shaped cells, the cocci or the spherical, and then we also have the spiral shaped cells as well. You'll notice some of the spiral shaped organisms here. This is an example of this direct immunofluorescent antibody, where you basically attach a fluorescent tag to an antibody, and those antibodies can adhere to the cell surface. So we're able to see how different microbes interact, where they are within tissues. When I was in graduate school back at the University of Rhode Island, I was in one of the lab groups that basically studied the way that probiotics work and came up with the methodology to how bacteria in the intestine behave. And it was termed, and it's now patented, it was called the restaurant hypothesis. And one of the things that we had to do was study the location of your normal flora bacteria versus your pathogenic bacteria in the intestine. So what we would do is we would use mice as a sample. We would actually extract their cecum, and we would use a cryogenic, basically almost like a cryogenic knife that would create very thin tissue samples. And through this staining process with these immunofluorescent tags, we would then be able to see within the mucus layer of the intestine as well as near the cell, they call them the villi, we were able to see where the different microbes were associated. So we could understand the mechanisms of E. coli intestinal diseases. So that's just an application of how this is used in a microscopic sense. We also can use isolation media. Obviously, selective media we've talked about allows us to be able to inhibit all of the microbes except the one that we're looking for. We also have the differential media, which will allow us through some type of a color change to see different bacteria on the same growth media. And then the biochemical testing. You'll notice this strip down here that has, this is basically a one long tool that has all of these different compartments with a different test in them. And basically what you would do is you take this needle with bacteria on the tip, and you would actually push it through all of these compartments to inoculate the compartment with a sample of the bacteria. And this would allow you to conduct like 20 different biochemical tests looking at the metabolic pathways that microbes use. So it gave you a great number of tests in terms of the physiological reactions that a lot of these microbes had. And then we have a series of miscellaneous tests. So we already talked about phage typing, which allows us to distinguish bacterial strains. So things like Staphylococcus and Salmonella. Remember that when we talk about strains, if we're looking at the hierarchy of how things are organized or classified, we go from kingdom to species, species being the most specific. Well, in order for us to distinguish very subtle differences between species, we can do what we call looking for strains. So phage typing allows us to be able to distinguish between those bacterial strains, those subtle differences in bacteria like Staphylococcus or Salmonella. We can obviously inoculate animals. We can do an in vivo test where we inject and look at how the disease presents itself in animals. And then obviously antimicrobial sensitivity. We can use things like the Kirby-Bauer test, which uses the discs on the plate, or an MIC to understand the types of antimicrobial drugs that impact microbes, and also what concentration resists growth. So a lot of the tools that we use as microbiologists to unpack these test results come in a multi-series set of almost like encyclopedias. It's referred to as Berge's Manual of Systematic Bacteriology, and it's a basically 5-part series, and it has every known and studied bacterial cell in it, and it has all of the different test results for that microbe. And one of the ways that they organize it is through what's called a dichotomous key. And dichotomous means that there's two different routes that you can take. So for instance, if I do my gram stain, which as we've mentioned is kind of the first test, and I notice that I have a bacilli, I have a cylindrical cell, I can then determine, is it gram positive or is it gram negative? Does it stain purple? Does it stain pink? So let's imagine for a second that it stains pink. Then I'm going to follow this track. Then I need to know, how does it use oxygen? Does it respire oxygen, or is it impacted by oxygen? So is it aerobic? Is it an anaerobe? Um, so in this case, if it was aerobic, if it had the oxidase enzyme, and it wasn't curved at all in shape, then that would tell me that it's either the genus Pseudomonas or the genus Alkalinogenes. So this is a really important tool that takes in a lot of these biochemical and phenotypic characteristics and allows me to get a pretty rough determination of what species I am working with. So we've already talked about kind of the breakdown, the cocci being the spherical. So oftentimes we look at things that are either catalase positive or catalase negative. Remember that catalase is an enzyme that breaks down toxic oxygen radicals into things like oxygen gas that are safe and non-toxic for microbes. So obviously, if we have clusters, we always talk about the staph. If we're looking at chains of cocci, we're looking at things like streptococcus or enterococcus. So this flow chart is going to allow us to get to this minute detail. And then obviously we have our gram negatives. We're talking about things like Neisseria. And then obviously we can do the same with the bacilli too. We know that our endospore formers are things like Bacillus and Clostridium, very small group. So you know right away that if you have an organism that produces endospores, and remember that when we do the endospore stain, the spores stain green in color due to the malachite green, and the vegetative or normally growing cells, the rod-shaped cells, stain pink due to the safranin. So we get these green endospores in this kind of hue of pink vegetative cells. And that would limit us to things like Bacillus or Clostridium, which are from the soil. If they're non-endospore formers, then we need to understand, are they acid fast? Do they have that waxy cell wall made of the mycolic acid? So that would be things like Mycobacterium. And remember that the acid fast stain, any of the acid fast cells are going to turn that pinkish purple color. And then obviously we can break down the gram negatives by whether they're aerobic or they're facultative anaerobes. And even further, if they ferment glucose, then we know we're in one of those groups like E. coli or Enterobacter. So when we get into genotypic methods, which is our second category, This is using genetics. It's using a very specific, precise technique that only requires very small amounts of a sample. And again, remember that these molecular methods do not require growth. You don't need to grow them in the lab to study them. We talk a lot about the probes. So we come back to this probe concept. And we come back to the concept of, I should say, hybridization right here, which uses these small pieces of DNA. So you'll notice here's the small piece of DNA, which we know is a probe. And it has all these little bases, these A's, T's, C's, and G's, sticking off of the kind of this side of the ladder. So think about this as a zipper on a jacket. So if you think about your zipper, you've got kind of the little metal pieces in the middle, and then the zipper actually rides up and down and either opens or closes the two sides. Well, that's kind of how hybridization works. If these two sides match together, then they're going to stick together perfectly, almost like when a zipper on a jacket closes. If they are not closely related, then that hybridization is not going to take effect, and you're not going to get this attachment or hybridization to occur. So the probes have a little fluorescent marker on them that is going to allow us to be able to visualize these connections under the microscope. And we've talked already about fluorescent in situ hybridization, which uses a fluorescent molecule. And we can also use what's known as electrophoresis, which is another technique that is pretty common and separates DNA pieces based on their size. So we can separate larger fragments out from smaller fragments. We've got the PCR, or polymerase chain reaction. We mentioned PCR earlier. It's like a molecular Xerox machine. It's going to allow us to use a probe to make very large numbers of copies of a very small piece of DNA. So it's a very sensitive and specific reaction. And we use it to detect things like HIV, Lyme disease, HPV, tuberculosis, and hepatitis are some of the big tests that we use this for. And then obviously we can sequence. We can take things like DNA and RNA, and we can understand the series of bases that comprise that organism's genetic material. So here's our electrophoresis. Again, we're separating out all these little bands that you see here represent pieces of DNA. Larger pieces of DNA move much slower, so we would load all of our genetic material up here at the top in these little wells. We would run an electric current through the samples, and these pieces of DNA would migrate down the gel. So the ones that get closer to the bottom are smaller in size. The ones that are larger move slower and will stay up towards the top. And what we can do is actually compare different DNA samples to see if they match. You'll notice that all of these here, 1, 2, and 3, have the same kind of banding pattern. Number 4 is entirely different. It does not have the same banding pattern as the first 3. We also have the immunological methods, and we talk a lot about serology, which is the study of the molecules in the serum. Specifically, your antibodies. Antibodies are derived from B cells, and antibodies are produced for a response to a specific infectious agent. So your antibodies match to a foreign substance, which we refer to as an antigen. And this antibody antigen reaction that occurs is very specific, like two puzzle pieces or a key into a lock. In order for this antibody, which you'll see here, it's kind of represented as this Y-shaped molecule, to fit to the antigen, which looks like this little X or almost like a jack, for these two to fit together, it's like a lock and key. Very specific reaction. So we can understand if you have antibodies in your serum to a particular infectious agent. That gives us an indication as to whether you've ever been exposed to that infectious disease. So again, we mentioned it's not only a very specific reaction, lock and key, but it's sensitive. It requires only a very small amount of an antigen for a reaction to occur. So we talk a lot about two different tests. We have what's known as agglutination, and agglutination uses whole antigens. So basically large intact antigens. And what we basically do is we affix an antigen to some type of an inert particle. So something like a latex bead. And that allows us to be able to see the interaction between antibody and antigen. The other side of that, we have what's called a precipitation reaction. And that's where we use soluble antigens. So something like, for instance, the VDRL test, which is used to diagnose syphilis. It can be either done in on an agar plate or in a test tube. And basically what you look for are these cloudy or opaque zones, which indicate where that antigen-antibody reaction occurred. So when this, here's our antigen here, when we get this kind of cross-linking of all these antigen and antibodies together, it forms this kind of clumping reaction, which gives us an indication of a positive result. And it's usually a visible sign. In this case here, it may be a color change or clumping, something that's visible to us to tell us that this reaction between antigen and antibody is occurring. So again, we've talked about this clumping, or it looks almost like bubbling that's occurred, indicating that the antigen and antibody have had a reaction. Again, just some other pictures showing you some different examples. We just talked about agglutination, which is an antibody reaction to a whole cell antigen. So we basically take the entire whole cell, we mix it with an antibody, and we look for the presence of this antigen-antibody complexes that form, generating this clump. And this clump is insoluble, so it basically settles out of the solution. So we do agglutination a lot to do blood typing. ABO blood typing is done through agglutination. And many bacterial and viral diseases are characterized through this mechanism as well. And then we also mentioned the precipitation tests, which are either done on the surface of an agar plate or a test tube. And what we would do is we would put our antigen in the middle. We put our serum samples on the side, and then we also have a control antibody over here. And what we do is we look for those cloudy, opaque bands that form, indicating that there was an interaction between the antibody and the antigen, giving us kind of that positive result. And we mentioned the VDRL test, which is done to diagnose syphilis. We also talk about the Western blot. So the Western blot is a confirmatory test for things that are like HIV. So typically what you would do first is what they call an ELISA test. And we'll talk about ELISA in a little bit. But to confirm that, we would do what's called a Western blot. And a Western blot is, again, it's a type of electrophoresis. We're running electricity through this gel. We're loading our sample towards the top. And what we're doing is we're separating proteins. So instead of genetic material, we're separating proteins in a Western blot. And we do what's referred to as like an immunoelectrophoresis. And this is where the serum proteins are separated out by the electrical charge. And then the antibodies specific for serum proteins are placed in kind of this parallel trough, and it forms these little reaction arcs for each protein. So we are able to conduct tests that involve like monoclonal antibodies, and that improves the specificity of these immunoelectrophoresis tests. So the sites of the specific antibody binding will appear as these kind of band patterns in the gel. So this is the confirmatory test to diagnose HIV. We also talk about complement fixation, and this is a test that is done through hemolysis. So we're lysing open cells. So again, a lysin is an antibody that requires the lysis of that antigen cell before the reaction can occur. So basically, what happens is purified proteins are added to a tube containing the antigen and antibody complex. The antigen and antibody react first, and then we usually add cheap red blood cells to the mixture. So this is used to diagnose many viral, rickettsial, and fungal infections. And again, we've talked about we test that antigen antibody reaction, and then we mix in those cheap red blood cells. So if when we're looking for basically whether there's hemolysis or no hemolysis as our reaction. So this kind of walks through in a pictorial format what you're looking at. So if we're talking about stage 1 here for a positive patient serum, we have the antibody and antigen complex that form first. The complement is then affixed to that antibody. And from there, we then add in the sheep red blood cells and we look for whether we have a reaction. So do the red blood cells lyse or not lyse? So if there's no hemolysis, we know there's an antibody present. If there is lysis, then we know that there's no antibody present. We also have several other serological tests. We have one that's specific for looking for syphilis, which is called the TPI test. We can also attempt to neutralize a toxin to determine what type of toxin is produced, letting us know which microbe is present. We can serotype, so we can actually type for different species and strains. This is useful for identifying different serotypes of streptococci. We also have what's referred to as the quelling test. And the quelling test allows us to serotype the capsular polysaccharide of pneumococcus. So this is just another type of test that allows us to identify a particular type of organism. And then obviously we have in vivo testing. So we're able to introduce antigens directly into somebody's body, such as the tuberculin test, which is designed to let us know if somebody was exposed to tuberculosis at some point. What happens is you get this raised red induration at the site of the inoculation, and it's usually greater than about 15 millimeters in size is the cutoff. And that lets us know that somebody was exposed at some point to tuberculosis. Doesn't necessarily mean that the person has an active case. Same kind of test is done with allergy testing as well, where we inject a little bit of the antigen under the surface of the skin and look for, a rash and puffiness. We can also we've mentioned the fluorescent antibody testing. That monoclonal antibody is labeled by a fluorescent dye. We can look at direct testing, where we take an unknown test specimen or an antigen, we fix it to a microscope slide, we attach a fluorescent antibody to it, and it helps us to identify the antigens that are on the surfaces of the cells. So we do a lot of direct testing to diagnose a whole slew of diseases like syphilis, gonorrhea, whooping cough, and meningitis. Our indirect testing is where we then would take the fluorescent antibodies and we react them with the portion of another antibody. They call it the FC, the constant portion of the antibody. So when you're looking at an antibody, we have this Y-shaped pattern. The top portion of the Y is what's known as the variable region. The bottom, or the tree trunk, is known as the constant region. So that means that in every antibody, this bottom portion is always the same. It's the variable region on the top where the antigen binds that is considered the one that's different. So the indirect testing allows us to diagnose syphilis and viral infections as well. And basically what we do is we take this secondary antibody, we attach it to the primary antibody, and if there is a reaction where the antibody binds to the antigen, this secondary antibody is going to glow. If these two don't interact, antibody and antigen don't match, the tag doesn't glow, we don't get any fluorescence. That tells us it's a negative result. So we're going to talk about ELISA. ELISA is one of the tests that behaves in this way. They use a secondary antibody. ELISA is a test that when there's that antibody-antigen complex that's formed, there is a colored product that is produced by an enzyme reaction. And most times we use what's known as the indirect ELISA. We'll talk about that in a second. So the ELISA uses a colored agent. Similar to ELISA, we have what's known as radioimmunoassay. And this is where instead of using a colored product, we actually use a radioactive material that would emit a glow to it. So indirect ELISA works this way. One of the things that we do is we attach an antigen to the bottom of these wells on the surface of a plate. And then we can add in the antibody. And this antibody has a secondary antibody attached to it with a fluorescent tag or a tag. And if there is a reaction where these are matched together, when we create an enzymatic reaction, it's going to create a color change in the wells. So we're going to actually get this kind of purple color that will form in the liquid. And this is basically a way for us to know. And the way we would read it is if we mixed in the antibody and the antigen and there was a reaction, that would tell us that it's likely that there was the presence of antibodies in the patient's serum for that infectious disease. So again, this is a presumptive test. It doesn't confirm. We'd have to do a Western blot in this case to really confirm that that is the truth. So again, we would add an antigen to the wells on a plate. We would then add in an antibody. And if there is a reaction, if the two come together, we would add our secondary antibody. The secondary antibody binds to the constant region of the first antibody. And then we basically use an enzyme to create a reaction. And the enzyme only creates the reaction if this complex is formed, the antigen and antibody have come together. And basically what's going to happen is it's going to turn this colorless liquid in the well to a purple color. And that's going to tell us that there is the presence of an antibody to that infectious agent in the person's serum. So here's an example of this microtiter plate where we've attached to the bottom of these wells an antigen. We've added in the antibody, and anywhere that there is a enzymatic reaction, we get this color change, which indicates that there is the presence of antibodies to that infectious agent. The capture or antibody sandwich ELISA method, basically the difference is that the antigen is trapped between two antibodies. And this is often used, it's a variation of the indirect method that's used to detect measles. So Lastly, let's talk about how do we diagnose viruses. And this is really, really, really important. Remember, we cannot grow viruses on traditional laboratory media. You are not going to cultivate these things in vitro in a test tube or on a petri dish. Viruses have to be grown using either a cell line or a tissue sample. It has to be injected into an embryo or an egg, or there has to be direct testing on an actual living animal. Viruses are dependent on the host cells. So, what are some techniques that we utilize to diagnose viral infections? Well, obviously, we would observe a patient's signs and symptoms. We could then take a series of samples from the patient and do a microscopic examination. And again, we can't use light microscope because it doesn't have the range to be able to detect something that small. So typically when we're looking at viruses, we're using the scanning electron microscope. We already talked about we can cultivate them using egg samples or tissue lines or cell lines to do so, some type of a living system. We can also do a series of direct observations of viral infected cells using that fluorescent antibody test as well. We can also do a serological analysis. We can detect the antibodies to a virus in a patient's serum sample. And then lastly, we could also amplify that viral nucleic acid and do a genetic analysis, study the actual hybridization or the genetics of the virus. So this is really important to make sure that you understand these are several of the techniques that we use to diagnose viral infections. So this is just a slide that kind of walks through these here in terms of signs and symptoms, microscopic examination using the scanning electron microscope. That is it. That wraps up chapter 17. Feel free, bring your questions to the office hours as we get into exam 4's material. Let me know if you have any questions on anything, and I look forward to seeing you all soon. Take care.